My name's David DiLillo. I've just wrapped up my postdoctoral studies with Jeff Ravitch at the Rockefeller University, and have just started as a scientist at Regeneron Pharmaceuticals. So I think some of the most interesting work is, again, as I mentioned, the ability of us to harness the power of the immune system to target a tumor cell and kill it in a relatively rapid fashion. So, for example, we've found new ways to stimulate long-term cellular immune responses against tumors through the use of antibodies. And so for the longest time, antibodies have been in the clinic for 25 years almost, but they've mostly been used to just target the tumors themselves. At this point now, we're using antibodies to target the immune system to either turn off regulatory pathways that are suppressing immune responses that can be unleashed to target tumors, or to stimulate immune responses against tumors themselves. Some of the studies that we've recently published, we've been able to show that you can use an antibody to trigger an immune response against a tumor, and this powerful long-term memory T-cell response can generate long-term memory against the tumors and prevent rechallenge of, for example, experimental animals with subsequent rechallenges of tumor cells. The future of the industry is bright. I think right now we're on the cusp of some incredible discoveries and development of new therapies that are really going to make impact on the lives of patients. And in my focus, we focus on cancer patients, and the new therapies that are just beginning to be approved and are in development seem to have incredible potential, mostly in the arena of harnessing the power of the immune system to turn it on the cancer cells where it had previously been turned off in order to eradicate the cells from the patients. So again, in this immuno-oncology field that has been explosive so far, I don't think this is and I hope it's not a short-term fad. I think that there's real potential here to expand. The knowledge that has been gained in the immune system over the past 20 years has really allowed us to identify these regulatory pathways that turn off immune responses, and if we can turn those immune responses back on, we can stimulate immune responses. And at the same time, we've identified pathways that can be agonized through the use of antibodies to more potently stimulate immune responses against tumors. So for example, CD40 antibodies, when you cross-link CD40 on antigen-presenting cells, you can actually trigger potent T-cell immune responses against tumors and wipe out the tumors in experimental models. And we're hoping that through the use of Fc engineering to modify and enhance these antibodies, we can get much better efficacy in vivo. I think when we talk about these immunotherapy approaches, some of the biggest challenges might be toxicities. So when you unleash the power of the immune system, it can there's a reason these regulatory pathways exist, and it's to prevent tissue damage against yourself. So when you unleash these pathways, different regulatory checkpoint cascades, the adoptive transfer of chimeric antigen receptor T cells, the use of bispecific antibodies that bring together T cells and tumor target, you can get significant toxicity. And I think understanding how we can limit those toxicities, either through engineering the antibody or the affinity of the antibodies, would be really important to look at. In addition, I think it's going to be really important to understand the differences in tumors from patient to patient, right? So of course, no tumor is the same, and so no single therapy would work as just a blanket therapy. So really understanding how each person's tumor is different from one to another to understand what therapy would be ideal for those patients. So for example, using various biomarkers, you can go after the easy ones like PD-1 and PD-L1 expression, but I think we're going to have to get much more detailed analyses of the patient's tumors to understand which antibody therapeutic and which checkpoint blockade or agonistic antibody would be ideal for their case. And it's going to take, I think, some significant time and significant sequencing and in-depth finding of biomarkers to be able to do this. I think it's, as I mentioned, I think it's happening. I think it's this growth of the immuno-oncology space and using the power of the immune system to eradicate tumors. As far as a specific target or specific molecule, it's tough to say in the next 6 months in the short term. But over the long term, multiple classes of antibodies that target regulatory pathways, and of course the chimeric antigen receptor T cells. Maybe one of the things that we could see in more short term would be novel combinations of these therapies. So, we're seeing that a lot of these checkpoint inhibitors and agonistic antibodies, they work okay on their own, but maybe in combination that they can be a synergistic effect. So maybe in the next 6 months to a year, we'll see some exciting results using combinations. I know it's being worked on intensely by multiple groups.